When you move, the thousands of microscopic sarcomeres inside your muscle fibers rapidly pull together to generate mechanical force. This action is executed entirely by proteins. These large, nitrogen-containing biological macromolecules operate as the functional workforce for virtually every physiological task in the human body. They build structural tissues, catalyze metabolic reactions, transport oxygen through the blood, and neutralize invading pathogens. Yet this immense functional diversity is constructed from a remarkably small inventory. The human body generates all of these complex structures by combining just 20 standard building blocks. By examining how these 20 amino acids link and fold into specific hierarchies, we can track exactly how systemic metabolism works and precisely where clinical pathology begins. Dietary nutrients supply the raw materials, but the resulting proteins act as the strict mechanistic blueprints that dictate health and disease. At the core of every protein is a single amino acid. This molecule is built around a central alpha carbon, flanked by a nitrogen-based amino group on one side and a carboxyl group on the other. This foundational backbone remains completely uniform across all standard amino acids. The distinct identity of each comes entirely from its variable side chain, the R group. Its specific geometry and chemical makeup dictates whether the molecule carries a charge, dissolves in water, or repels it. Humans require all 20 variations to function properly, but the body can only synthesize a portion of them from scratch. The rest must be absorbed directly from our diet, making them nutritionally essential. The essential amino acids required from the diet are identified using the mnemonic PVT-TIM-HAL: phenylalanine, valine, tryptophan, threonine, isoleucine, methionine, histidine, arginine, leucine, and lysine. A few specific amino acids dictate distinct physical behaviors in a protein chain. Glycine is the smallest, allowing it to pack into extremely tight spaces. Proline has a rigid ring structure that physically forces sharp bends and kinks into the chain. Cysteine contains sulfur, allowing it to form strong, covalent disulfide bonds with other cysteines to lock a structure into place. Even a microscopic alteration in a single R group changes the molecule's chemical reactivity. The physical properties of the protein at the monomer level strictly govern how it will behave in the body. To build a functional protein, these monomers must polymerize. The carboxyl group of one amino acid reacts with the amino group of the next, forming a rigid covalent connection called a peptide bond. This condensation reaction forces the removal of a single water molecule, which locks the connection in place. As this process repeats hundreds or thousands of times, it creates a long, continuous sequence of amino acids. This linear chain is the protein's primary structure. The primary sequence dictates everything that happens next. The precise order of the R groups determines exactly how the entire chain will fold upon itself. The chain begins to collapse into localized, two-dimensional folding patterns, establishing the secondary structure. When the sequence coils into a spiral, it forms an alpha helix. Hydrogen bonds between the amino acids secure the turns of the coil, a structural pattern highly prevalent in keratin. Alternatively, sections of the chain can align side by side to form a flat beta-pleated sheet, providing high tensile strength to structural tissues. These predictable local folds form the necessary transition step, converting a simple one-dimensional chemical string into the foundation of a complex mechanical tool. The secondary structures then fold further, condensing into a complete three-dimensional architecture known as the tertiary structure. This is the protein's final functional shape. This exact geometry is held tightly in place by four key molecular interactions: hydrogen bonds, ionic interactions, disulfide bridges, and the clustering of hydrophobic R groups deep within the core. Many physiological tasks require multiple proteins to work in tandem. When separate polypeptide chains assemble into a synchronized unit, they achieve quaternary structure. Hemoglobin operates precisely this way, requiring exactly 2 alpha and 2 beta chains bound together to capture and transport oxygen. A protein's ability to perform its job depends entirely on maintaining these precise folds. When an environmental stressor disrupts this geometry, the protein undergoes denaturation. Extreme heat, highly acidic or alkaline conditions, alcohol, and heavy metals all possess the chemical energy to break apart the delicate bonds holding the fold together. Crucially, denaturation destroys the secondary and tertiary structures, resulting in an immediate loss of biological function, but the primary peptide sequence remains entirely intact. Biological function requires a stable three-dimensional architecture. If the specific fold fails, the physical mechanism fails with it. Before the body can utilize these structures, it must break down dietary proteins into their constituent parts. Digestion begins in the stomach, where hydrochloric acid denatures the food, and the enzyme pepsin cleaves the large chains into smaller peptide fragments. The process moves into the small intestine, where pancreatic enzymes like trypsin and chymotrypsin systematically dismantle the remaining chains. The final absorbable products are free amino acids and short peptides, which cross the intestinal wall into the bloodstream. Once inside the cells, these amino acids are repurposed. During transamination, an amino group is transferred to a keto acid, synthesizing a completely new amino acid. Clinically, every transamination reaction strictly requires vitamin B6 as an enzymatic cofactor to proceed. However, breaking down amino acids poses a direct chemical threat. Oxidative deamination in the liver removes the nitrogen-heavy amino group entirely, generating free toxic ammonia. The human body cannot tolerate circulating ammonia, as it rapidly causes neurological damage. To neutralize this threat, the liver employs the urea cycle. This metabolic pathway captures the free ammonia and binds it into a safe, water-soluble urea molecule. The blood then transports the stable urea directly to the kidneys, where it is filtered and excreted. Systemic metabolism is a continuous balancing act. The body constantly breaks down and rebuilds proteins, harvesting their utility while meticulously preventing nitrogen from reaching toxic levels. When the primary sequence of a protein contains an error, the systemic consequences are severe. Sickle cell disease demonstrates exactly how a single molecular substitution dictates clinical failure. The pathology originates in the primary structure. In the beta-globin chain, a polar gluconate molecule is accidentally replaced by a nonpolar valine. This single hydrophobic shift causes the entire quaternary structure of hemoglobin to mutate. The chains aggregate together, physically pushing the red blood cell membrane into a rigid sickle shape that clogs capillaries and triggers systemic ischemia. Similar principles govern protein misfolding diseases. When normal tertiary structures misfold, they create toxic protein aggregations that deposit in tissues, driving the pathology behind amyloidosis and Alzheimer's disease. A failure in protein intake is equally destructive. Severe protein energy malnutrition causes kwashiorkor. Patients presenting with kwashiorkor suffer from extreme dietary amino acid deficiency. Without the building blocks to synthesize necessary transport proteins, fluid leaks into tissues, resulting in generalized edema, liver fat accumulation, and severe skin changes. By contrast, marasmus involves a total caloric deficiency, forcing the body to break down its own structural proteins for basic survival, causing profound muscle wasting. From the precise sequence of peptide bonds to the rigid requirements of nitrogen clearance, structural chemistry forms the basis of all physiological reality. Proteins are the biological machinery that drive cellular function. Understanding their exact structural hierarchy is a prerequisite for understanding the root mechanisms of human disease.